Terrific. Well, good morning, everybody. My name is Kevin James. I'm Violet's dad. And when I'm not Violet's dad, I also teach at the University of Virginia. And one of the things that I teach students about at UVA is biology, okay? The biology of cells. And I have heard that everyone here has learned some things about cells over the past couple of weeks, right? I think we have a big project coming up soon. I have the project in the back of my car. That's going to be the next thing after I get done with this. And so what I thought I would do is tell you a little bit more about cells, tell you some new things that you haven't heard before, but that'll build upon the things that Ms. Oley taught you about over the past couple of weeks. And it's going to involve a switch, if you will, from cells in general to cell types. And I'm going to speak more about what I mean when I'm talking about cell types in just a moment. But first, I've also heard you've been working on some Word Masters challenges. Okay, also been working on that last night. And so I'm gonna ask you to remember one word that you probably haven't heard before, but is important for the class today. And that word is differentiation. Can I get a repeat of this? So differentiation. Although I will coordinate this. Differentiation. Differentiation. Terrific. Okay. And let's see. What else do we need to do? It's a noun. All right. Its root is differentiate. Okay, so that's a verb. And there are a couple of different definitions, but I'm going to talk about the biological definition of differentiation. And that is right here, the process by which cells change from generalized kinds of cells to more specialized kinds of cells. And analogies will follow. All right, so what I want to do is first I'm going to show you a picture of a generalized, generic animal cell. Looks familiar, right? I think you have a picture of this in your notebooks, right? Color-coded and so forth. We have nuclei, we have lysosomes, Golgi bodies, plasma membrane, and so forth. I know of no cell that looks like this, okay? This is equivalent to me saying that a fifth grader looks like this. This is not too bad to me. This looks kind of similar to over on the left-hand side here. How many people have a head? How many people have two arms? Okay, two legs? Great. So those are all up here. Okay, but let's ask some more questions. How many people have hair? I still have a little hair left over, so I'll still raise my hand. But how many people have blonde hair? Not as many hands. How many people have long blonde hair? Even fewer. Okay. How many people have facial hair? Okay. Specialization. Okay. Little bit different, all right? These are the things that make us individuals, individualized, and cells individualize also. And that process, in broad strokes, is called differentiation. Here are the types of differentiated cells that I know, okay? These ones look very different than the ones that show up in the notebooks, but these are the ones that are more reflective of the types of cells you encounter in your body, specialized parts of your body, okay? And so what I'm going to do is talk briefly a couple of different cell types that you might find interesting. There'll be some movies, there'll be some jokes. Please laugh. And then there's going to be some activities on the computer for you guys to go hunt around and see some cells in not your own body, but in another human body. And you can see all different shapes and patterns and things that they exhibit in our bodies. Okay? Any questions up to this point? Certainly raise your hand and things like that. I got tons of time allotted for people to ask questions. Yes? Is the one in the top right a nerve cell? It is a nerve cell, and I have a whole slide dedicated to neurons in just a moment. All right, off we go. All right, so the first cell type that we're going to talk about are called the neutrophils. I'm pretty sure that most people have never heard of neutrophils before. Neutrophils are really interesting because they're really weird, okay? This is what a neutrophil looks like in cartoon form, okay? See that multi-lobed blobby thing in the middle? That's the nucleus, okay? Yeah, and it's all weird shaped, right? Not a nice little circle like that jelly bean image that I showed from before. And this is one of the characteristics of neutrophils, such that if you take one of these spheres and imagine you cut it midway through, it'll actually look like it has 2, 3, 4 nuclei because you're cutting through that weird shaped nucleus. The other thing that's characteristic, a form of specialization or differentiation of neutrophils, is that they have lots of these things called granules. You see these specks here inside the cell, okay? Those are like some of the vesicles and Golgi bodies that I think you have in your notebooks, but they have a much more specialized function that I'm going to show you in a moment. Okay, what do neutrophils do? Well, what their job is, their specialized function, is to chase down and kill bacteria. The way that they kill bacteria is by releasing those granules inside. So all those little speckles in there, inside they contain lethal compounds for bacteria, okay, that get released when a neutrophil encounters a bacterium. One at a time.
Does that keep them alive, that stuff?
Does it keep them alive? Well, it keeps...
It says that they kill the bacteria and then die.
Well, so when they release those, it is a very toxic event, so much so that the neutrophil dies as a result of killing the bacterium. Neutrophils in our body only last about 24 hours, so they live fast and die quickly. So we are constantly turning over neutrophils in the body. There's a question over here. Yeah, I'm going to repeat the question for the video. Are they a type of white blood cell? They're kind of like white blood cells. They exactly are white blood cells. They're one example of a white blood cell. There are other types. Oh, there are multiple types. I could give a whole semester's worth of information on white blood cells. This is just one example. Yes? Kind of. Well, let me show you a video how they do it. Okay, there's an element of that. And you're going to see what happens where the dead ones go in just a moment. This is a very old and classic video. All right, I want to highlight 3 things to preempt any questions. First is the neutrophil, an activated neutrophil, is this guy here. All right, so it's not circular like in the cartoon. It's spread out flat because it's crawling along a surface. This little brown speck over here, that's a bacterium. Okay, so that's something that you're going to see it jittering around. It can move and so forth. All the other spheres here are other white blood cells, could be other dead neutrophils or other things. They're just like balls that are in the way of the video, so they're not going to be doing anything. You want to pay attention to the chase between this neutrophil and the bacterium here. And it's pretty cool. And so what that neutrophil is doing effectively is sniffing out the bacterium and is pushing the other cells out of the way and going after that bug. Going after that bug, very persistent. Okay. Chasing, 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 and eventually it catches it. Okay, and then you can't see that in this video, but this is when you will have this process of degranulation, the release of all of those granules. Reactive oxygen species, all this nasty chemistry, kills the bug, and eventually will kill the neutrophil. Yeah, Mia. Great question. Why, if it releases those granules, doesn't it do damage to neighboring cells and things? The compounds that get released are so reactive. It's a chemical reaction that gets done. They're so reactive that they have already done their chemistry within a very short distance of the cells. And so the damage happens. That's why the neutrophil can't avoid the damage, because it's released it. But the neighboring cells they aren't damaged. But if there's something really, really close by it could happen. You could do a little bit of collateral damage. Yeah? Excellent question. Yeah, so you astutely noticed that there seemed to be another bug there that the neutrophil ignored because it seemed to be chasing the other one. Recall what I said about sniffing out the bacterium. There's actually a chemical with a trace that it follows. It's like perfume almost, okay? And so if somebody's sort of sitting there and not really moving, there's not as much motion of that perfume. Okay, but the bug that's chasing away you kind of can smell it, and it smells like it's stronger over in that direction, and that's the trace that the neutrophil is doing. It's like a bloodhound too, sort of sniffing out the case. Yeah.
So basically, this reminds me of like those Roadrunner cartoons. This time the coyote actually catches it.
Correct. Yeah, there's a little bit of the Roadrunner and coyote. Hopefully, the coyote wins most of the time in these battles. Yeah.
In the video, it was like way bigger than the actual bug. Why does it have to release the granules if it can't just like absorb it or something?
Oh, excellent question. It said, well, look, a bacterium is only about, well, I'll give you some numbers here. A bacterium is only about 1 micrometer, so that's 1/1,000,000th of a meter in size. A neutrophil is 10 micrometers, so 10 millionths, right? So it's 10-fold difference that you see here. Why doesn't the bacterium just scoop it up? Why doesn't the neutrophil just scoop it up and eat it? Sometimes it will, and there are other white blood cells that only do that. They don't do the degranulation. They just do this process of engulfment, and they just eat them up. And so they will do that as well. But sometimes there, for example, are too many. Say there are 1 million bugs in that vicinity. This is when you want to do that collateral damage on everything else. You go in there, you just blow up everything that you can. You try to eat as many as possible, but any ones that you don't eat, you destroy in the area. And then was there one more question over here?
So the neutrophils are sort of like kamikaze superhero bloodhounds of the human body.
Something like that. If that's what has it, yes, the kamikaze bloodhound superheroes, if that's what you remind of them. Well, actually, I'm going to get to the superhero part of this in a moment, because they're not done. Okay? Sometimes neutrophils can't catch the bugs. And then if they don't, what they will do is eject. It looks like they're barfing out their DNA. Remember, the DNA is in the nucleus? Okay? So here, this is like Spider-Man. Okay? So squid out a web of their DNA from their nucleus and use it as a net. They're actually called nets to trap bacteria in the web. And as they squirt that out, there are some of those toxic compounds on the web that then will go and degrade the bacteria. This is only like 10 years old. I can show you what it looks like. So this is what happens when they can't find the bacterium, but they're activated and they're looking for it. This is a different microscopic view, circles and things here. Those are your neutrophils, and they're going to add something in that activates them. You're going to see they all spread out. That was the addition, and then a couple of minutes, and then now, where's the bug? Where's the bug? Where's the bug? But there are no bugs to be found because they just put in the whiff, but they didn't put in the bug, and now they're all activated. And I think that the movie will stop because you can't see that web there here with another. If you look by fluorescence, those are those webs of a couple of the neutrophils squirting out their DNA. Okay, here's a better example. We did better at imaging them. So here now neutrophils are labeled fluorescently in red, and DNA is labeled in green. So look at the green. It's the same type of experiment. So they're going to get activated, and then trying to squirt on, I think. Okay. It's pretty cool, right? Well, on the topic of that, so here's your analogy. We already learned that neutrophils are white blood cells. Live neutrophils ordinarily circulate in our bloodstream. Any guesses on where we might find dead neutrophils? Everywhere. Well, no, not everywhere. It's not. Okay, when you blow out your nose, all that green and yucky stuff, that's mostly dead neutrophils. Yeah. So the reason why it's a different color is because those reactive chemistries there that I talked about before, they change the color of the red blood cells. So they react with the red blood cells and they change them a different color. Yeah. How did they get into your nose? Yeah. So they're in the blood, and we have all of our tissues have blood circulating through them. They will exit the bloodstream when they sense an infection. If they catch a whiff of one of those infections, they will squeeze out of the circulation and go into our tissues, like the mucous membranes that are in our nose, and then fight the bugs there where they reside. And then was there one other question over here?
So is that when you're sick, there's more bacteria, so there's more than neutrophils to put on your nose?
Yes, and so we have many of these types of defenses, but this one's really potent against the bacterium. Correct, all those things coming in and doing this chemistry and things. All right, we're going to do one more so I can get through my other 4 cell types. Yeah? Mm-hmm.
Do allergies have a very similar thing going on?
So the question is about allergies. Allergies involve the immune system. However, they involve very different categories of white blood cells than these. These ones, there are some other disorders that can mean when you have not enough neutrophils or too much activation. Allergy is another class of white blood cells. Cool. All right, so we're going to move out of the immune system, now out of the blood, and into another specialized cell type called an adipocyte. Adipocytes have a very interesting differentiation characteristic. They are characterized by one big fat vacuole called the lipid droplet. Okay, adipocytes are fat cells. So if you remember, that's what it kind of looks like. Okay. And so people are about fat. Okay, so if you see fat at the butcher or something like that, it's all white and things. Those are the adipocytes. Okay, and this is what they look like. What you might not know is that not all adipocytes are the same. In fact, there is even a specialization, a differentiation that can occur among adipocytes that do really different things. The fat, the adipocytes that you see at the butchers, okay, those are called white fat cells, white adipocytes. These are the ones that are important for storing fat, right, in that lipid droplet. Okay, so we eat too much or there's too much fat circulating in our bloodstream, adipocytes will uptake those molecules and store them in that chubby vacuole. But in fact, we have another kind of fat called brown fat that does something completely different. It is a source of heat generation when we're cold. And this was also something that was rather new. It wasn't known that we had this. We knew that mice and rodents and bears and things had this other type of brown fat. But what we have here, it's in weird places. It's not in our bellies. Okay? It's all up around our collar here. And you can show it by different imaging techniques. So here's a person here, normal temperature. There's no contrast. You don't see anything. But then if you put that person in the cold and you image them just the right way, all those dark areas there, that's areas of brown fat that are revving up their metabolism. They're generating heat. It's like a little internal stove. They're using those fat stores, okay, to keep us warm, all up around this area. And as I said, mice and rats, bears, mammals have these, but they're hidden in different places, but they're thought to serve this function. Yeah? Does it explain warm-blooded animals? No, not entirely, but it can, what it does refer to is some of the adaptations of warm-blooded animals to help maintain their temperature when it gets really cold. So it's like a version 2 of that to help that be preserved through evolution. Right, and so you could think about why it would be advantageous to have that heat generation all up and around here. It's like a fat scarf, okay, right around the neck. Absolutely.
So
Yeah.
that's why the warmest areas when you're cold is when on your head and your chest and
Well, for some of it here in and around the neck. The head is for a different reason. The head is for because you have a lot. We talked about the bloodstream and things. You have a lot of blood vessels up and around your head, and so if those are exposed, we don't have a fat head. It's sort of skull and a lot of vessels, and so that's why the heat escapes off the head. How do you know which one is white and which one is brown? Very good question. Yeah, the colors are different. Yeah, so it's not very imaginative. So white fat, you know from the butcher is white. Brown fat, if you isolate it, you can see it as a different color. If you look at it under the microscope, it's really a different color. And you also see the cell organization of brown fat. Not quite so chubby. They organize a little bit differently in the tissues. They have a certain shape that's characteristic of them. So there are different ways. And there are also proteins and molecules that brown fat express that white fat don't. And so that we'll circle back to that near the end because you're going to look at some markers of different cell types. Okay. All right. Out of fat and into another cell type called fibroblasts. Okay. Fibroblasts, there's an image of a fibroblast, generally really stretched out, a lot of cell membrane processes and so forth. Fibroblasts make the protein glue that holds us together. Okay, so if you say we're a big bag of cells, or if we're a big bag of cells, why am I standing here? Right? We would just be a bag of cells it'd be like a lot of little tiny marbles. We'd all be on the floor in a pile. Okay, so we have glue, we have proteins that cells stick to, and that those cells, those proteins stick to one another, and that's what enables us to hold our shape. Okay, and fibroblasts are critically important for creating that glue, maintaining it, turning it over, particularly when we get hurt. How many people have a scratch? Yeah, even I have a scratch. I cut myself trying to repair our laundry dryer last night. When you get a scratch, okay, there's the bleeding, and then there's the scab and things like that. But how many people, if you skin your knee or something like that, all of a sudden it feels like really tight when it's healing, right? You can't bend it and things like that. That's because of fibroblasts. Okay, and I'm going to show you. Let me show this video, and then then I'll answer questions. Okay, and you can actually mimic the process of wound healing in the lab through this experiment called a scratch assay. So I think there's audio here. There's a couple of fancy words in here that you don't need to know, but what you're looking at here is, imagine over on top a flat so-called monolayer, which means one cell layer of fibroblasts just sitting on plastic in the laboratory. And when they say confluent, all that means is full. All the puzzle pieces, there's no empty space, so they're totally full.
Fibroblasts grown in vitro in a culture dish
Culture.
form a confluent monolayer of cells. Cells in a monolayer are relatively static. Contacting each other inhibits their migration.
So that's like your skin, for
Some
example.
cell layers can be wounded experimentally by scratching them with a needle.
So you just skinned your knee. Here's the real experiment when you do this.
In such an experiment, we can observe that the fibroblasts at the edge of the wound become migratory and quickly move to repair the gap.
So this is what occurs underneath your scab.
It's important for wound repair in an intact organism.
And then they go and they fill in underneath. Now the reason why it gets tight is because fibroblasts are contractile. What they're trying to do, say in that wound experiment, you have a big gap and you have skin or skin type cells on one side and then the other side. They're coming in, and then when they find one another, they're going to start making some of that glue and pulling on it to try to get that wound to be sealed really, really tight. And so when your scab feels like it's really tight in the tissue there, if you have a scar, sometimes you'll see how the skin looks twisted or pulled. That's the action of the fibroblast, trying to bring those two ends together so that you don't open up another wound there down the road. Yes? If you have a piece of glass in there, I recommend you get it out, because the fibroblasts will not go through glass. But they can cover really large. I mean, I'm sure everybody's gotten pretty bad skinned knees and things like that. My recollection, I don't have all the statistics on this, but I know in your mouth, like if you cut a wound in your mouth, this happens at about 1 millimeter per day. So you can cover these cells can cover a pretty good distance in a pretty short period of time. Would a type of fibroblast be a platelet? No. So the wound healing process is like a two-step with platelets that are from the bloodstream. I didn't do platelets here because I figure some people have heard of platelets. Those are the ones that are important for forming the clot. So when they get activated and they form your scab, they're not really alive. They're kind of pieces of cells. But when they get activated, they release factors, kind of like the perfume that I talked about, but now is an attractant for fibroblasts. So when the scab forms, they release things that tell the fibroblasts to come in there. So you get scab first, and then the fibroblasts come in and will remodel all of the glue underneath so that you get the scar and the contraction and everything else. So the platelets in many ways hand off the wound healing process to the fibroblasts in the later stages of resolution. Yes, and so in this, when the scab falls off, you see newer skin. It's not only fibroblasts, but just the appearance of the scar, how it looks very different from the places where you weren't wounded. That is the consequence of fibroblasts in action, repairing that Yeah, I got a couple too. And then last one, and then we'll move on to the next cell type.
So, like, when you get like a bad scrape like that, it usually stays for like a really long time. Is like how long does it take?
Right, and so how big of the wound is and how long does it take to get better. You have two things to go in because it's just like this experiment. There's a couple things actually. This is reminding me some more things to tell you. So if I take this experiment and I do a small wound, okay, another place where they're getting that attractant is from the fibroblasts on the other side, and so they sense a void, and then they get attracted to one another. Okay, but if I take my finger or something very large and I make a huge gash in the plate or on your knee, something like that, it's really, really far apart. This is where stitches come into play, okay, because it's too large of a void for the fibroblast to be able to sense the edges and close the wound. And so the stitches and things help to sutures try to bring those close enough together so you can get that cross communication and the wound healing. I forgot what was next. Here's your neurons. Okay, so let's talk about the nervous system a little bit, just briefly. Okay, neurons you can think of conceptually like the electrical circuitry of our body. Okay, these are the wires that connect our brain and our spinal cord to all of the muscles and organs and things in our body. Like that. All right. It's white. Okay. Did anyone else, other than somebody, chew the iPhone cord when they were little?
Yeah.
Yeah, there you go.
Chew my Game Boy cord for some reason.
It's not white all the way through, right? What's on the inside? Wire, the metal. Okay. Same thing holds for the axons. What the white is on the outside is insulation. Okay, it's the thing that protects the wire. And it's not only to protect the wire from you so you don't get electrocuted. It's also to help the wire conduct electricity from here to here. Because if it doesn't, it could go in here, right? Or it could just go out into the environment. Neurons have exactly the same thing. Okay, they have a very long process called an axon that's like the wire. Okay. And they're really long. Just as an example, our longest neurons, if you want to talk about who's the tallest in the room, neurons win, okay? The tallest neuron in your body goes from the base of your spine to the very tip of your toe. So for me, it's about 1 meter, one cell, one long axon. That's a really big transmission for a wire. And so therefore, it needs to be insulated. And the way that it gets insulated is from a special biomolecule called myelin. It's a kind of fat. All right, made by another differentiated cell type called Schwann cells. You see, I have them labeled there. These are myelin-producing cells that wrap around the axon, the long wire, and keep it insulated. So they're like the white on your iPhone cord. Yes? Oh, really? Oh, yeah, so the Schwann? Mm-hmm. Yes, and so the Schwann, yes, Theodor Schwann, named after these cells are named after the individual who discovered the cells. Yeah. Right, and so how do you, how do you find them out? I think that you're going to set me up perfectly for the next thing. Oh, no, I have one more. Let me get back to that because this is an open question that I'm going to talk about some things that are happening right now about how do you find cell types if you don't know what you're looking for. All right. Okay, I think this is the last one I want to talk about are another form of specialized cell called myocytes. These are really beautiful cells. You can see them over on the right-hand side there. These are the main cells in muscle. Myo, muscle, heart, skeleton, okay? In your vessels of your, in your circulation have myocytes in them. And the first question that probably comes to mind are, what are all these almost zipper-like regular patterns in them? Okay, it's very characteristic of myocytes. They're called striations. And where that pattern comes up is from a special repeating unit in myocytes called a sarcomere. And so this is one sarcomere. So those zippers that look up on the top here, each one of those is one of these units, blue to blue. And what happens is those zippers have these filaments. You see these red, they're called thin filaments. They're like forks here. And then in between the thin filaments are thick filaments. Those are the ones in green. And in the green, thick filaments have the equivalent of a rowing team on them. They have little paddles that will row through the thin filaments when the muscle cell needs to contract. I don't think this is a little bit too detailed for you all, but I'll show you the image. So if there's a signal from the nervous system for that muscle to contract, those green guys start rowing in between the thin filaments, and then they bring those two forks together. Okay? Repeat that on every single one of those zipper units in the myocyte. Okay? And now you go from a cell that's this long to one that's this long. And this is how myocytes contract. And we can do this in a dish also. So here are some. This is also on plastic. Okay? Muscle cells, given the right stimulation, will just start beating spontaneously in a dish. Okay? And that's all those sarcomeres coming together, relaxing, coming together, relaxing, coming together, relaxing. Exactly what happens in your heart. Those are heart-like cells. Yeah, and so if you take all of those in your skeletal muscle, say your bicep, right, you have a long linear array of these, just like in the image up on the top here. And if all their sarcomeres bunch together, well, that volume's got to go somewhere, so it goes in the other direction.
Does that have anything to do, like, if, say, you're paralyzed, does that have anything to do with, because you said it was.
Oh, if you can't contract? If you can't control your muscles?
Yeah, like, so for the last one.
Mm-hmm. Yes, and so yeah, your question is, I said that, for example, there's the neurons that go from the base of your spine to the tip of your toe. What happens if you can't control your legs? What has gone wrong? Is it a problem with the skeletal muscle? Is it a problem with the nerves? It is almost always, although not always, almost always a result of damage of that axon, either the one that goes to the tip of the toe or the ones in the spine that relay the information to the one that goes to the tip of the toe. Okay. So those are a couple of representative differentiated cell types. I'm in an engineering department, and we also teach math in addition to biology, so I like numbers. And so I'm going to give you a couple numbers about molecules, cells, cell types. All right, so the first thing is all that stuff, remember the DNA, the stuff that the neutrophils barf out and so forth, okay? We know exactly how many letters of DNA make up us. And the number is 3 billion, plus or minus, okay? So there's I am defined, at least genetically, by a 3 billion sequence, okay, so that's 3 with 9 zeros at the end, of these A, C's, G's, and T's, long stretch after another. We know this number, okay? We have all of the sequence. Now, we also know that those 3 billion letters encode information for genes and genes that make proteins. This is the stuff that happens in the nucleus and that impacts all of the other cells. We also know how many genes that there are. It's far fewer than we guessed originally, but the number is about 20,500. Okay? In fact, there is a water flea, okay, so a little thing that floats around in the ocean, that has more genes than we have. Yeah, so gene number, the number of genes that we have, doesn't give an indication of complexity. There are some plants that have way many more genes than us. So those letters define 20,500 genes. And then those genes are expressed in how many different differentiated cell types in our body? This gets back to the question before. How do you find these things? We have no idea. We don't know. I listed 5. We probably have documented 500, but we don't know how many there are in the body. And so here we're going to get some help from Facebook, of all places.
Yay.
Okay. So does the word Zuckerberg sound familiar? He's Mark Zuckerberg, yes. Okay, he's the founder of Facebook. Lots of money. Thankfully, he's decided to spend it on good causes, and some of these are encompassed in this initiative with his wife, Priscilla Chan, okay, to spend $3 billion. I don't know if 3 billion is for the 3 billion bases, the DNA from before, over the next 10 years to work on projects like this one, this so-called human cell atlas here. And the goal is to try to put a number on those question marks on the preceding slide. So it's a big group of scientists getting together, and they're going to try to mine different cells and tissues and so forth to see how many that they can find. Because in theory, we should know this number, and probably some of those cell types go bad in different diseases. They're important for certain processes that we do every day, but we don't know the answer. Okay. That just started. So I have no movies, no pictures, nothing really to show you except just the number, and that they're going to be working on it over the next 10 years. Maybe you see it in the news. Maybe by the time you guys are studying biology in college, you'll hear word on this. But what I can tell you is what we think that human cell atlas, so atlas like a map, okay, we want to get a map of all the cell types in the body, okay? If this effort is successful, what's it going to get us? I think that the leading thing that we're hoping to get out of that human cell atlas would be these things called markers of differentiation, okay? These markers usually mean a gene that is expressed or a protein that is expressed by a cell that gives an indication of the type of cell that it is, all right? As an example, we already saw some of these. Neutrophils exhibit a funny nucleus. They express those toxic compounds that make up the granules. Neurons have these long Schwann cells that wrap around on the outside. Myocytes have those sarcomeres, okay? These are all proteins that are expressed originally by genes, and that help enable us to define these different cell types in our body. Now, the cool thing is, I mean, we knew this in a general sense for a long time. And we already have many good protein markers to look at different cell types that we've known for decades in our body, in our organs, and we can look at them right now. And we actually are going to look at them right now? Yeah, we are going to look at them right now. And so this is where we're going to transition to a computer exercise. We're going to use another atlas that is already out and about. And it's called the Human Protein Atlas, which seeks to localize these markers and other proteins in all tissues in our body. And so right here, what we'll do is, are we handing out something, or? Everybody's in? Yep. Okay. All right, so everybody up here, I'm going to, the way that, as I said just a moment ago, what I'll do is I'm going to show you how to navigate through the site. And then I'm going to go back to my slides, and I'll give you example markers for neutrophils and neurons and myocytes and so forth. So then you can go hunt around and you can see some really pretty pictures of them and where they localize in the body. But before we do that, I want to just show the site so you can get oriented, and then you can hunt around and go wherever you like. The site is free, as you can see. You use it for teaching. You go home, you can hunt around and look at whatever you like. Okay, so the search starts from up on the top here. And what the website wants you to put in is a gene name, a protein name, a marker that they have in this big database of images for different cell types and tissues. And we had a question before, how do you find these things, and so forth? What you will be looking at are images of tissues from people, okay, that have been preserved in such a way. So imagine you have a block of tissue. So you have a block of muscle, something like this, okay? Now I'm going to take that block of tissue, And we were talking about the butcher. Let's talk about the deli counter now. You know those deli slicers, right? All right, so you take the tissue and you run it through a very expensive deli slicer, and you cut an extremely thin section of that tissue, okay? Tiny, like 1 cell thickness, okay? Then you put that onto a slide, and then we're going to look over on the top, and if the section is thin enough, we'll be able to see all the individual cells in that slice if we use certain reagents that enable us to give color to those protein markers. All right, so what you'll be seeing are a lot of circles, which are so-called cores. So imagine a hollow drill drills into that tissue, and then you cut the deli slices from there of different pieces of a liver, a heart muscle, a brain section, so forth. And then the brownness of the staining that you see, okay, that indicates where the protein is that we are searching for in this field up here. I'm just going to take one example here. I don't even know what CD36 is, but I know it will get us to this site here. Okay, so you're going to punch in the names that I will give you on the next slide. And the names that I give you are unambiguous enough that only one thing should come up on this list. But for this one, there's multiple ones. Here's this molecule here. It's going to tell me all these things about the protein. Here's the part that's relevant to you, is that what we have over here is this marker, how strongly expressed that protein is in different tissue types. And so, for example, if I go to, let's see, there was a liver here. Oh, there we go. Let's see. So we go to the liver. I click on that. It's going to give you more information. And then here are one of those deli sliced cores of the liver. And I can click on any one of these. And there are more examples up here. And you can zoom in. Okay? And so you can see, I should say, so the brown is that CD36 molecule. Those are the cells that are positive, that are marked with that protein. But you can see other cells here. See the nuclei, the blue circles? Okay, those are stained by another color. Those ones are negative for that marker. So they don't have CD36, but the ones right next to it do. And so you can hunt around, you can zoom in, you can click on another example. See, pattern's a little bit different. We can go to, I don't know, let's see. Some of these will look boring, or like in the brain, for example, where it's not really expressed. See, not really much anything to see. Negative, okay? So what I'll give you on the slide on the PowerPoint are some markers to look at and some tissues that give some really striking patterns for neutrophils and myocytes and neurons and so forth. But if you want to go off the beaten path and you want to try some other things and stuff like that, you can hunt around and look at different tissue types and so forth. But what I want to do is give you an appreciation of the diversity of cells that are in the body by looking at them directly on the computer.
Okay, so I know it's gonna sound really random, but as a point. So
I'm ready.
my dad has this weird eye marker. I think it's like genetic, but if I found out what that is, I could look it up and like.
Yes, yes, you could. If you knew that there was a certain gene or protein that you're really interested in, if they have the reagents. Now, this is a 15-year effort to go in and do this, and it's still ongoing. And we don't have good reagents, good ways to localize every single protein in the body, but that's the goal of this atlasing effort. And so many of them, you probably have about a 60% chance, 70% chance of having it in here, at least something provisionally in there. Okay? All right, so here are the best markers that I could find that give cool patterns for neutrophils, adipocytes, fibroblasts, neurons, and myocytes. Okay, so this all you would do is if you want to look at the neutrophil stain, you type FUT4 into that first field, and then the place where I saw the coolest staining for this is going to immune system and then looking in your tonsils. Okay, and so you can look at some people's tonsils and you can see the different patterns. And I picked that one because you should be able to see a couple examples of apparently multinucleated cells, and that's that funny nuclear shape of the neutrophil, so you can look around and zoom in. AdipoQ is for in adipose tissue. You'll see that staining S100A4 for the skin. MAP2 in the brain and hippocampus, and then this troponin T1, muscle tissues and skeletal. And so now I'm done talking. Hunter, you can do things out of order, compare notes with your neighbors, okay? And I'll wander around if people have questions and things like that as we go.
So you can type in any of these codes.
So we're going to log off and shut down. We're going to put our computers away. Make sure you are logging off and shutting down, not just shutting down. Then we're going to head back to our classrooms to grab our lunch and head to lunch. Before we do any of that, what do we need to do?
Say thank you.
Thanks, guys, for paying attention, asking lots of good questions. It was fun.